Please like, share and subscribe EduPhy India. Thank you. Class 12th Biotechnology. Separation and isolation of DNA fragments. Separation and isolation of DNA fragments. Now we can separate the DNA fragments by cutting of DNA by restriction endonuclease results in the fragments of DNA. So these fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through a medium or a matrix. So whenever we do perform gel electrophoresis, the charges are opposite of the separating particles and of the field. So if the particles are moving towards the anode or the DNA fragments, they will be moving towards the anode. That means anode is of positive charge and DNA fragments do have negative charge. And nowadays the most commonly used matrix is agarose, which is a polymer extracted from the seaweed. So agarose is a natural polymer. The DNA fragments separate according to their size through sieving effect provided by the agarose gel. Sieving effect, it's like a tube and the particles go down from upper to the bottom, and the particles smaller will be separating first and the larger at the last. In this way the DNA fragment separates or resolved according to their size. The separated DNA fragments can be visualized only after staining the DNA. Since DNA is very transparent and very thin, so it's important to stain it to get a colored DNA, and then it is visualized through the microscope. So to visualize, we use the stain ethidium bromide followed by exposure to UV radiation. And UV radiation is important because we can use the laminar airflow also, that is a machine, biotechnological machine that provides UV radiation. Obviously it's harmful, so precautions has to be taken, but we can't see these in visible light because DNA fragments ain't visible. So you can see bright orange colored bands of DNA in ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. Now we have the separate and pure constructed bands of DNA. This step is known as elution. So the DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Now in the figure you can see a typical agarose gel electrophoresis showing migration of undigested lane 1 and digested set of DNA fragments lane 2 to 4. So you can see the wells are made 1, 2, 3, 4, and these are the orange color bands. Orange is due to the ethidium bromide stain, and they are separated according to their size by sieving effect. And you can see the largest to the lowest. These largest and smallest are the DNA bands, and this process is gel electrophoresis for the separation of DNA fragments. Cloning vectors. Cloning vectors. What is cloning? The ability of making multiple copies of a vector genome in a host cell. Suppose our host cell is bacteria, and bacteria do have ability to make number of copies or replication of the DNA, chromosomal DNA. But whenever a bacteriophage or plasmid, what is bacteriophage? Bacteriophage is a kind of virus that attacks the bacterial cell. So whenever the bacteriophage attacks the bacterial cell, it makes the number of copies as similar as the bacterial cells. So bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. So bacteriophage made to have more capacity of multiplying even than the bacterial cell, but ever then if bacteriophage attacks the bacterial cell, so it have the ability to use the replication property and make multiple copies of the bacteriophages. So some plasmids are also have the ability to to make multiple copies and may have only one or two copies per cell, but others may will be having 15 to 100 copies per cell, and their number can even go higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number is equal to the copy number of plasmid or bacteriophages. So this technique is used for carrying out desirable genes, to gain desirable genes. We will see it further later in that same topic. So vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. Now the following different types of steps are required for cloning vectors. Here you can see the 4 steps you can see the here are origin of replication, selectable marker, and cloning site. See what happened. Origin of replication, this is the first step where the sequence from where replication starts, and any piece of DNA when linked to the sequence can be made to replicate within the host cell. So this sequence is also responsible for controlling the copy number of linked DNA. So first our step in cloning any gene, any vector is ORI, that is origin of replication. So in this a piece of DNA is linked to the host organism. Second is selectable marker. This you can understand by, if we want, as we saw that we need some desirable gene, but what we are getting some undesirable genes also. So because of recombinant DNA technology, we are able to get desirable genes rather than undesirable. So it was very easy, it became very easy. So in here same, while linking any piece of DNA, we need a selectable transformant. Now what is transformant? Just right from the definition of transformation, that is a procedure through which a piece of DNA is introduced in a host bacterium. So when we are trying to make it come, so this is the selectable marker helps the vectors for introducing them with transformants and eliminating the non-transformants. So transformants, what they do, are the selectable markers. They identify the selectively perfect transformant and eliminates the non-transformant. So this is the second step. Third is cloning site. So cloning sites, these are some recognition sites that vectors binds to. So this could be one or more than one. When it is commonly it is one, and if it is more than one, then it is complicated to link the DNA piece. So recognition sites for the commonly used restriction enzymes, presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance gene. So this is an example of cloning sites. For example, it is the ligation of alien DNA. Okay. Insertional inactivation. In case we took the example of antibiotic resistance gene, that helps in selecting the transformants, whereas the other antibiotic resistance gene gets inactivated due to insertion of alien DNA and helps in selection of recombinants. So as we saw, we need transformants when we are selecting the transformants from the non-transformants. So, whenever we are introducing any transformant, selecting the transformant, the antibiotic resistance gene gets inactivated due to insertion of alien DNA. So, when this is inserted, the process is insertional inactivation. A recombinant DNA is inserted within the coding sequence of an enzyme beta galactoside. So this is the example of insertional inactivation. Next is vectors for cloning genes in plants and animals. Agrobacterium tumefaciens, that is pathogen. Pathogen that is introduced in the dicot plant that transfers tDNA. The normal cell of dicot plants is converted into tumor, and tumor makes a chemical for pathogen. So this Agrobacterium tumefaciens, it is a pathogen that enters into the dicot plant. That is giving the dicot plant tDNA that introduced into it. The normal cell of dicot plant is converted into tumor. Tumor cells then makes a chemical that is useful to this pathogen. Next, retroviruses in animal transform normal cell into cancerous cell. So retrovirus, again, it infects a animal and the cells of animals becomes cancerous, and obviously in both the cases you can see the plants and animals are facing harm from the pathogen as well as from retroviruses in plants and animals respectively. These pathogens or viruses are now disarmed. How they are disarmed? By using the cloning sites technique, and where they whenever the cloning vectors have been changed or disarmed, that is, if the chemical from the Agrobacterium tumefaciens is removed out the retroviruses, the content of the virus, that is, if it is the removed, the replication, that could be, it could be having genetic material RNA or DNA. So whenever it is removed and the useful DNA or RNA is introduced in both of the cases, then they will be giving us the desirable genes in both plants and animals. So these pathogens or viruses are now disarmed and is used in ligating desirable genes into plants and animals. So once a gene or DNA fragment has been ligated into a suitable vector, it is transformed into a bacterial cell or animal host, and then where it is multiplied. So this could be used in bacteria also. Competent host. Competent host, it is for transformation with recombinant DNA. As you know, DNA is a hydrophilic molecule. It cannot pass through the cell membrane. Why? In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up DNA. So it is really easy to say that we introduce the alien piece of DNA into the bacteria as the host, but it is rather take multiple steps to make it. Okay, so one is competent host, where bacteria are made to first to be competent to take up DNA. This is done by treating them with a specific concentration of divalent ion. Divalent ion, cation, that is like calcium. So that increases the efficiency with which DNA enters into the bacterium. So in first method, cells are incubated with recombinant DNA on ice. So this is first method of making DNA to enter into the bacterium. So in which recombinant DNA can be forced into cells, such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius. a heat shock. This is a heat shock, and then putting them back on the ice. So this is a process of we can say, first the treatment is hot, then suddenly a warm, that makes a incubation process. So this enables the bacteria to take up the recombinant DNA. Next technique is micro injection. This is very easy. Recombinant DNA is directly injected into the nucleus of the animal cell. So in this method, it is suitable for an animal cell. Micro injection is good for animal cells. Then next method, it is generally used in plant cells, is biolistics or gene gun. When it is said biolistics or gene gun, that means firing shots or bombardments. So when cells are bombarded with high velocity microparticle of gold or tungsten, already in one experiment of atom we have studied about the about the atom gun. We have shown that electron gun is just pulled into the particles, alpha particles emitted on the foil. So in the same way, in biolistic there is gene gun where the cells are bombarded with high velocity of microparticle of gold with DNA coating. So they will be having DNA, and that helps to make them enter into the bacterium cell. This is useful in the case of plants. Please like, share and subscribe EduPhy India. Thank you.